Cell sorters are some of the most high-precision instruments in modern life sciences. They're used across research labs, pharmaceutical cleanrooms, and biologics manufacturing to isolate and separate delicate cell populations, often for high-value, time-sensitive workflows. But there's a problem. Cell sorters are complex systems with internal tubing, flow paths, and aerosol management systems. And that complexity makes them exceptionally prone to trapping contamination, especially in hard-to-reach internal areas. Cell sorters are critical in workflows like purifying cell therapy products, where only viable, healthy cells can move forward for patient use, or removing dead or damaged cells during vaccine development, where purity directly impacts accuracy and performance. In these environments, contamination isn't just inconvenient, it's expensive. It can lead to lost batches, delayed timelines, and costly shutdowns. Most facilities rely on manual cleaning, spray and wipe. But manual disinfection simply can't reach the sorter's internal chambers and tight tubing. Even when the outside looks clean, internal bioburden can remain. CDC-cited research has shown that even thorough manual cleaning may leave behind up to 60% of bioburden. Some facilities try to solve this by using more aggressive sterilants like formaldehyde or chlorine dioxide gas. But those options introduce a different risk: equipment damage. BD has explicitly prohibited formaldehyde and chlorine dioxide on certain models, including the FACS Discovery S ATM, due to material compatibility concerns. So the challenge becomes clear. How do you achieve full decontamination without destroying a high-investment instrument? CURIS Hybrid hydrogen peroxide vapor provides high-level disinfection without compromising sensitive equipment. Using a 7% hydrogen peroxide formula, CURIS HHP delivers greater than 6-log sporicidal reduction while maintaining safer conditions for internal components. And it's not just a claim, it's validated. CURIS was tested and validated for decontaminating the BD Fax Discovery S 8TM cell sorter inside a Baker biosafety cabinet following NSF ANSI Standard 49 Annex 2. Validation highlights include 15 test cycles using CURIS Trinity and Curoxide, biological indicators placed in 14 or more hard-to-reach locations, a 100% inactivation rate across all indicators, no observed impact on equipment performance or materials. This protocol is now recognized as a validated option for BD cell sorter decontamination. Compared to high-concentration peroxide systems, CURIS lower concentration vapor reduces common damage risks, including gasket cracking and seal degradation, residue buildup in filters, damage to sensitive electronics, and long off-gassing times. CURIS cycles are engineered specifically for material compatibility, making them ideal for cell sorters, biosafety cabinets, and cleanroom robotics. In the validated setup, the BD cell sorter was enclosed inside the Baker cabinet. CURIS vapor was delivered using preset automated cycles. Airflow and temperature were monitored, and biological indicators confirmed complete coverage, even in sample chambers and exhaust filters. This is full decontamination where it actually matters, inside the system, not just on the exterior. Can you use hydrogen peroxide on a BD cell sorter? Yes, if it's low concentration, contains no additional active ingredients like heavy metals, and is validated for use. CURIS meets these standards and is endorsed for this purpose. How do you decontaminate a cell sorter without damaging it? Avoid corrosive gases and use validated vapor systems designed for sensitive equipment like CURIS AHPTM. Is it compatible with biosafety cabinets? Yes. If you're relying on manual cleaning alone, you're leaving risk inside the instrument. CURIS gives you a safer, validated way to fully decontaminate cell sorters, backed by data, manufacturer validation, and smarter automation. Upgrade your decontamination process without sacrificing your equipment. Schedule a demo or request a quote today.